Stem cells. We're going to talk about the genetic stem cells, done by Hala, who's going to talk about genetic stem cells in general, and conclude. Layan, who's going to explain the two types of stem cells. Marah, who's going to summarize the whole thing in Arabic. What are stem cells? Stem cells are cells that are undifferentiated, meaning they do not have a specific job, function, or structure. Stem cells do not have the potential to become all other kinds of tissues or cells in your body. Stem cells are distinguished from other cell types by two important characteristics. First of all, they are unspecialized cells capable of renewing themselves through cell division. Second, they can be induced to become tissues or specific cells with their specific functions. Like for example, some organs such as gut and bone marrow, stem cells regularly divide to repair and replace worn-out or damaged tissues. That's how the body uses stem cells to replace worn-out cells when they die. Genetics, heredity, stem cells. Traits are mostly controlled by dominant alleles than recessive allele, because a dominant trait is more likely to show up than a recessive trait since a dominant trait is a stronger trait. The alleles of two parents combine to express them in an offspring by randomly inheriting a trait from each parent.
Related to what my partner defined and explained, I'd like to talk about the two types of stem cells, which are adult blood stem cells and embryonic blood stem cells. What is an adult blood stem cell? Like all stem cells, which shares at least two characteristics that have been identified in many organs and tissues, including brain, bone marrow, blood vessels, skeletal muscle, skin, teeth, heart, gut, liver, and ovarian epithelium. First, they can make identical copies of themselves for long periods of time. This ability to proliferate is referred to as long-term self-renewal. Second, they can give rise to mature cell types that have characteristic morphologies and specialized functions. The adult blood stem cells occurs in many tissues, and it can differentiate into specialized cells in tissues of organs. Next, it also can transdifferentiate into specialized cells characteristics of other tissues. Finally, could be used in clinical trials in patients who are suffering from myocardial infarction.
Hematopoietic stem cells give rise to all the types of blood cells such as red blood cells, B lymphocytes, T lymphocytes, natural killer cells, neutrophils, basophils, eosinophils, monocytes, and macrophages. They can differentiate into neural, cardiomyocytes, and liver cells.
What is an embryonic stem cell? Embryonic stem cells are derived from embryos as a developmental stage that are 3 to 5 days old. They are harvested during a process called in vitro fertilization. This involves fertilizing an embryo in a laboratory instead of inside the female body. Embryonic stem cells are known as pluripotent stem cells. These cells can give rise to virtually any other type of cells in the body. The process of making embryonic stem cells. First, DNA from adult cell injection into fertilized human egg with no nucleus.
2. Eggs begin dividing, forming a mass of cells. 3. After 5 days, it forms a blastocyte, a hollow embryonic ball of cells.
4. Stem cells are harvested embryo destroyed. What are the unique properties of all stem cells in general? To sum up, stem cells are capable of diving and renewing themselves for long periods. They are unspecialized and they can give rise to specialized cell types.
As well as, stem cells are a powerful tool because they have the potential to treat wide range of diseases, such as damage by the spinal cord, stroke, and Alzheimer's disease.
وبنختم موضوعنا اللي اخترناه عن الخلايا الجذعية بتقنيات وظروف الخلية الجذعية المستمدة. طبعا بيتم استمداد الخلايا الجذعية جينية من كتلة الخلايا الداخلية للجنين اللي طبعا بتكتسب من الحيوان الأم المانح. وذكر مارتون وماثيو التقنية اللي بتأخر بزرع الجنين وبتسمح للكتلة الخلية الداخلية طبعا بالزيادة. تشمل هاي العملية عدة خطوات. أولا إزالة مبايض الأم المانحة، من ثم بتتداوى بجرعات مع البروجستيرون، وبالتالي لما بتتعالج بتغير ذلك بنية الهرمون وبيئته، بيؤدي لحتى تبقى الأجنة حرة في الرحم لمدة تقريبا 4 إلى 6 أيام من هاي الزراعة. طبعا هاي الزراعة لازم تكون داخل الرحم، وبيتم حصاد الأجنحة وبتزرع في مستنبت المختبر حتى تشكل كتلة الخلية الداخلية هياكل بويضة طبعا مثل الأسطوانة، وبتنفصل لخلايا مفردة من ثم بتطلع على الخلايا الليفية المعالجة مع الميتوسين سي حتى تمنع الانقسام الفتيلي في الخلايا الليفية. من ثم بيقوم بإنشاء خطوط الخلايا المستنسخة عن طريق إنماء خلية واحدة. وأظهر إيفان زوكوفمان إنه الخلايا اللي نمت انطلاقا هاي الزراعات إما ممكن إنه تشكل ورم مسخي وأجسام مضيغة الشكل، وبتتميز بالمختبر وبتشيل كلها إلى الخلايا المتعددة القدرات. وبعدين استمد غير مارتن خلايا جذعية جينية وزرعها بشكل مختلف عن هدوك، بحيث قام بإزالة الأجنة من الأم المانحة حوالي تقريبا 76 ساعة بعد التزاوج، وزرعها خلال فترة الليل في الوسائط اللي كانت عم تحتوي على المصل. باليوم التالي قال إنه إزالة هاي الخلية الداخلية من أواخر الكيسية الإريمية طبعا باستخدام الجراحة المجهرية. الكتلة الخلوية الداخلية المستخرجة واللي تمت زراعتها بالخلايا الليمفية تعالج مع الميتوسين مثل فوق وفي الوسائط اللي تحتوي على المصل، وكانت مشروطة من قبل الخلايا الجذعية الجينية. بعد أسبوع تقريبا نمت هاي المستعمرات الخلايا، ونمت هاي الخلايا مزرعة الخلايا وأظهرت خصائص متعددة الإمكانات اللي دلت على قدرة تكوين ورم مسخي، وتميزت بالمختبر وشكلت أجسام مضغية الشكل. وبعدين بالنهاية أشار مارتن أن هاي الخلايا كانت بتعبر عن الخلايا الجذعية الجينية اللي هي موضوعنا اليوم.
To conclude, during the project and whole unit, I learned a lot about genetics. I became aware of stuff that I knew about but never knew how was it related to genetics. This was one of my favorite projects since it included a lot of interesting points that grabbed my attention. Here are some resources we used to get the information from. Thank you.